and COST-EOS as well as Marie for inviting me along. It's been a very interesting couple of days so far, seeing how a lot of these activities are coordinated in the European sort of zone. We don't necessarily have the diversity of modelling groups or countries in our region, yet we sometimes struggle with the coordination, so it's been quite interesting. So thanks to Marion for a very nice sort of introduction to the different spatial scales and time scales that emerge in biogeochemical modelling. What I'll be focusing on are the downscaled properties in the BGC, and you can sort of see from these sort of two pictures here from the Great Barrier Reef that a lot of the processes that occur in the coastal zone have much shorter length scales and much shorter temporal scales. So we're working in the order of sort of tens of metres up to tens of kilometres, and at time scales of a few hours through to days. So we're at a point where we're resolving tidal activity in these zones. So whilst I'm the person up here talking today, this work has been sort of undertaken by a very large group of people. Mark Baird and Matthew Mongin have been very closely involved in this. But the actual group that have contributed to this is very large. So our group at CSIRO has physical oceanographers that work on the hydrodynamic modelling. We've got Nugsar, who is in the sediment dynamics. There's a fairly large biogeochemistry team. I sort of sit at the intersection of the observations and the modelling in the data assimilation component. And I have to say the glue of the group and those that are often not well sort of noted are those in the scientific computing space, because a lot of what we do is very heavily reliant on software engineering. So a lot of what I'll talk about today, whilst I focus on the GBR, the Great Barrier Reef, there's similar problems that occur worldwide. So some classic examples of where downscaled BGC processes have helped explain dynamics for occurrences like the green tides that affected the Qingdao region during the Olympics. We see that harmful algal blooms are becoming more and more prevalent in certain areas. So, for example, NOAA now have a harmful algal bloom system, and Marion touched on that this morning and the way in which they go about predicting that. And then the other aspect being ocean acidification is starting to have direct impacts on coastal industries, of which oyster farmers on the west coast of the US have been impacted on this recently. So these biogeochemical processes, whilst there's global connections in them, they have very local impacts, and we're not immune to that in Australia, being a very large island nation. 97% of our population is on the coast, and I would say it's in our national psyche that we really consider ourselves a coastal culture. And when we start seeing impacts of changing ecosystems, for example, the oysters that are produced being affected by harmful algal blooms, whereby paralytic shellfish poisoning toxins are present in the oysters. That ends up having then economic consequences and flowing onto societal consequences as well. Other aspects being, last year was a particularly warm year when we look at the global heat content, and we're starting to see the impact of these elevated heat contents at the benthic communities. So along the west coast of Australia, we had a very large die-off of macroalgae populations, and that is possibly going to result in a regime shift and have impacts then on the lobster fishery. And in the GBR region, there's been substantial levels of coral bleaching this year, and that was predicted at least 6 months out by NOAA. And their suggestion was that these coral bleaching events that were likely going to happen during this austral summer were not just going to be confined to the Great Barrier Reef, but were going to be pretty much international in nature. And this has unfolded, and this was predicted 6 months out. So when we sort of throw all this into what we're trying to do here, is we're trying to look at what are the key processes and dynamics that are driving these processes, and how can we go about predicting that. So in terms of the way we've gone about that in our group, we use a nested modelling approach. So we've got a global model here, and the global model provides information about large-scale circulation patterns and connections, but not necessarily the very specific knowledge needed for particular management scenarios. So what we do is we have a series of nested models that go from, say, the 10 kilometres down to 4 kilometres, and we call that 4 kilometre model a bridging model. And that's so that we don't exceed a nesting ratio of 5 to 1, which is a commonly used rule of thumb. Then within the 4 kilometre model, we have a high resolution 1 kilometre model that is more suited for resolving certain processes. And then there's a series of estuary and bay models which have resolutions in the order of 100 out to 500 metres. So in terms of actually using the GBR as a case study, it's a very tricky area to model in that we have a north-south extent of around 2,000 kilometres, so it's a very large area, and we've got very significant inshore tidal zones. And within this area, there's perhaps only 16 or 17 regularly monitored in situ stations. So if we were trying to derive information from in situ data alone, it becomes very difficult because there's large spatial gaps in our observing capability. On top of that, the wet season up there, which is in the austral summer, ends up resulting in very large areas of cloud cover, so we're not actually able to derive a lot of the information that we might hope to from remote sensing. So in terms of trying to tie together all these processes, we've got a model that does that, and it needs to be able to resolve adequately over 3,000 individual reefs, take into account a diversity of corals with 600 different species, have 28 large rivers that flow into it, and be able to essentially model the dynamics of the large inshore seagrass beds. And the key thing about this is there's very many important BGC cycles which are location dependent. For example, we don't expect seagrass to be in water depths more than about 30 metres. So the model that we apply there needs to have a full gamut of processes in there to be able to adequately answer questions at the appropriate scales. So what I'll do is I'll step through the value of downscaling in terms of what does the spatial and process complexity add. Go through how we undertake a model assessment, the impact of data assimilation in this reasonably complex model, and then present a tool that we use to actually upscale local studies to whole of system characteristics, and then we'll try and link the modelling activities with actual management decision making. Okay, so process representation. So this is your classic intermediate complexity pelagic biogeochemical model. You can think of it as having a phytoplankton component that's linked to a nutrient component. We then have zooplankton and detritus, and each one of these arrows here represents a process that transfers mass between each of these components. This model is represented by a set of ordinary differential equations that are evaluated on a cell-by-cell basis, and these components are then advected and diffused around the system by a hydrodynamic model that's forced by meteorology. We have this sort of funny carbon chemistry component that sort of sits in between the two models, where we're allowing atmospheric fluxes to change the in situ DIC and the like, and that carbon chemistry component is quite important for reef processes. So the additional level of complexity comes into account when we need to move into the coastal zone. So when we go into the coastal zone, we need to include a benthic component, and our benthic component has coral, seagrass and macroalgae in it. And we also have a sediment model. So the sediment model simulates the way in which sediments are introduced into the model via rivers and then redistributed via sediment resuspension. Now, the other key difference here is each one of these orange components here is an optically active constituent, and this becomes important a little later when I introduce the optics model. So this model is now running in near real time. It's been running in near real time for the last 5 years and produces daily output of pretty much all state variables and then a set of derived variables as well, like degree heating weeks for coral bleaching. So as I mentioned, this model has got an optics component to it. It makes it somewhat unique in that it's one of the few biogeochemical models out there with an optics component. And you might ask, why is this important? So in the first case, we know that different wavelengths of light are attenuated at different rates. And whilst that might not be so important in the blue water, it is particularly important in coastal waters where we've got sediment resuspension and CDOM. So this is a mechanistic optical model, but it's somewhat simplified compared to full radiative transfer models like Chl-a. So this model was recently published by Mark Baird, and when we look at the error budgets in the optical model, we see that the error in the model, in the actual optical model, is small, but the mismatch between optical measurements and those predicted by the model are largely dominated by errors in the modelled constituents like phytoplankton concentration or sediment concentration. Okay, so let's have a look at what the effect of resolution is. So I'm going to use the 4 kilometre model here as a coarse resolution model, and we'll use the 1K model as the high resolution model. So what we've got here is the difference between what the 4 kilometre model grid cell bathymetry is and the maximum depth for that cell as predicted by the very high resolution bathymetry data set that was used to generate the coarse resolution model. So you can see that generally in the GBR lagoon here, the errors are relatively small in the order of maybe 5 to 10 metres. But where we've got very topographically complex areas like fringing reefs, for example, we see that the errors are quite large and may exceed 50 metres. If we now compare that to the 1 kilometre model, we see that the error in the bathymetry is substantially reduced. So what's the actual impact of that? Well, if we look at an example like Barrier Reef, which is in Heron Island, we see that with the GBR1 model, the depth at this particular reef is 3.7 metres, but the depth from the GBR model is 13.4 metres. And that has an immediate impact on the way in which heat, for example, is introduced into the model through diurnal heating. And we see that the amplitude in the diurnal heating of the GBR4 model is in the order of 0.3 degrees, but in the GBR1 model, we've got 0.8 degrees. Okay, so there's an immediate effect on process representation when we look to higher resolutions. Second example being, if we look at the distribution of nitrate in the GBR4 model, so this is moving inshore, we see that in the coarse model, nitrate concentrations on the fringing reefs sit between about 2 and 6 milligrams of nitrogen per cubic metre. And the inshore, where we're introducing nutrients through a river plume, that nutrient distribution is very localised around the mouth of the river. So then if we move in to have a look at the high resolution model, we see that because we're resolving the reef more adequately in this region, and because we've got a tidally resolved model, the tidally induced pumping of deep water up into the surface layer brings nutrients to it. So we start to see that the distribution of nutrients in the reef is more represented. Second being that the actual river plume, rather than being constrained to this coastal embayment here, is allowed to propagate more appropriately along the coast, and we see the plume dynamics improved by the 1 kilometre model. Now, what's the impact on the phytoplankton then? So this is the total chlorophyll A as predicted by both the models. First of all, we see that the chlorophyll A is largely constrained to the reef here. But if we look at the GBR1 model, we see far more fine-scale structure emerging, and that ties back into the talks yesterday when we saw high-resolution examples of NEMO and RECOM shown. We start to see that sub-mesoscale structure allowed to propagate in. One thing I'd like to point out, though, is whilst we might allow that fine-scale structure to exist, we can't necessarily constrain it adequately with assimilation yet, because our observations aren't necessarily able to constrain these very high these very short spatial length scales. Okay, so moving into the sediment component. So the sediment model allows for a couple of processes here. We have the advection diffusion of suspended sediments, but there's also a settling and flocculation process, and sediment is then allowed to accumulate in the top 3 layers of the sediment. And there's a couple of processes that then act in the sediment bed. First of all, there's consolidation, which means that the longer the sediment stays in the sediment bed, the more cohesive it becomes. And the second aspect is there's bioturbation there. So we know in many sediment classes we end up with lots of bioturbation, and that mixes the nutrients in terms of the distribution of pore water. Next being we were able to include benthic plants. So this isn't necessarily important in deep water, but certainly in the coast where we've got optically, where the optics allow light to penetrate to the bottom. It allows a couple of seagrass species to grow, so Zostra and Holophila, and they grow by uptaking nutrients from the sediment, and there's an above ground biomass and a below ground biomass. So this is particularly important for answering blue carbon questions. When we actually look at the distribution of seagrasses predicted by these models, generally it's whilst we might allow the seagrass to be initialised at a certain depth zone at a constant value, the emergent properties of the model and the associated mortality rates of the seagrass only allow the seagrass to survive at certain areas where they're able to grow, and that's largely determined by the light climate. Next one being the carbon chemistry and coral modules. So the coral is important when it comes to the dissolved organic carbon and inorganic carbon, and it represents coral host growth, the zooxanthellae growth, and the associated chlorophyll with the zooxanthellae. And the calcification dissolution processes are also important. So by taking into account the carbon chemistry in the EREFS model, we've been able to come up with pretty much one of the first estimates of a whole of GBR risk, well, exposure to ocean acidification. So here we're looking at the difference between the aragonite saturation on the reef versus open water. And you can see at the offshore reef edge, the aragonite saturation is close to ocean values or slightly above due to calcification dissolution. But as we move across the reef into the coast, we see the aragonite saturation being substantially less than the open ocean. So by having a mechanistic model of the carbon chemistry, we were able to then tease apart the actual drivers of what's changing that aragonite saturation. So this is particularly important for trying to get a handle on which reefs are likely to be able to be protected by intervention and change by human activities versus which ones are either buffered or protected by the influx of oceanic waters. So given that the processes that I've presented now cover a whole range of I suppose bioregions within the model. If we now focus on the pelagic component of phytoplankton growth, we realise that still it's a reasonably complex model, and unlike the hydrodynamics which are built around the Navier-Stokes equation, many of the phytoplankton growth equations are somewhat empirical in nature. So with that in mind, we know that the model is particularly sensitive to a couple of parameters. So in the past we've undertaken parameter perturbation experiments to get a feel for how sensitive the model is to particular parameters. And what we see that is if we introduce around 50% uncertainty in a number of the phytoplankton and zooplankton parameters, we end up with an order of magnitude uncertainty in the phytoplankton and zooplankton state. So the other thing to note here is that some parameter combinations end up causing the model to go into an unstable zone, and therefore those particular combinations should be avoided. And this really sort of raises the point that at some level there needs to be some effort globally, and we know this is taking place, that appropriate priors should be constructed for putting prior information across these parameters, because we know that they are physiologically based in many respects, and that if we can come up with appropriate priors for these parameters, this will enable us to reduce some of the uncertainty in the model prediction, especially when it's coupled to a parameter estimation or state estimation procedure. Okay, so one kind of concept that I think is worthwhile considering here is that there's many studies that are undertaken in these coastal environments, especially in reef environments, that look at processes at individual reefs. So there's no way that these processes and studies can be extrapolated to all reefs using observation campaigns. So one of the tools that we've come up with within the eReefs project is called RECOM. So RECOM is a tool which allows managers or experimental scientists and observationalists to go into a web-based interface and come up with a grid that's appropriate for the particular application that they're interested in. And it's a very easy workflow. So you log into the RECOM web browser, you define a grid, select a time period that you're interested in, and then it gets the appropriate boundary conditions from the particular model that we'll be forcing with, which is either GB1, GBR on an FTP server that you can download. So a lot of the RECOM infrastructure is built around the Marine Virtual Laboratory infrastructure, and there's a reference to that there if people are interested in it. But what does that actually enable you to do? So recently we had 2 French summer students come and work with our group, so Elisa and Marion, and they put together very high resolution models of 30 of the reefs within the GBR region. So these are the 30 reefs that are sort of scattered so that this reef number 1 and 2 correspond with these ones down here, and reef number 22 and 17 are these ones up here. And looked at the aragonite saturation of these reefs over a period of time. And this allowed us to look at what the impact of OA was at reef scales that were unresolved in both the GBR4 and GBR1 models. So each of these reefs take approximately half an hour to construct using RECOM. So we've done 30 reefs with 2 students so far. So we were wondering if we can upscale this by having a number of other students in the future. But down the line, we expect that such a tool, instead of being sort of applied by a human user, could be automated. And the idea behind this is that these models will subsequently be two-way coupled with the GBR1 model to allow fine-scale reef processes to propagate back into the coarser resolution models. Okay, so undertaking an assessment of how well these models perform. First of all, I'd like to point out that in this region, the satellite-derived chlorophyll A is particularly unreliable. So if we plot in situ chlorophyll A against the Modus OC3 chlorophyll A, we get errors up to 300% in a lot of these regions. So whilst there has been efforts to go about generating locally specific algorithms, they're still sort of somewhat unreliable. So using our optics model, we can actually predict a simulated OC3 or simulated GSM chlorophyll, if you like, and compare that against the simulated surface chlorophyll A. And what we see that is using this optics model, we see that there's immediately a bias in the IOP-derived estimates of chlorophyll A. So this is sort of represented schematically here by this relationship. So satellites actually sense the top of atmosphere radiance. There's then some function that's applied to that through atmospheric correction and some in situ, well, some in-water estimates as well to give you a remote sensing reflectance. And then that remote sensing reflectance using empirical studies is then often related back to chlorophyll A. So it's very hard to generate an error budget using this method. So the alternative is, given that we have a system state here in an optics model, we can use that to predict a simulated set of IOPs, a simulated set of AOPs, and then from that simulated set of AOPs, we then derive a simulated remote sensing reflectance at the surface. So this is spectrally resolved. We can do it at any of the wavelengths that are currently observed, whether it be Modus or VIIRS, or perhaps more excitingly, we can potentially do this at the RGB colour bands that drones may be able to observe as well. So if we build in this optics model, it opens up a whole range of possibilities that aren't available to us if we neglect biooptics. So here's a comparison of the observed reflectance at 488 nanometres against the modelled reflectance at 488 nanometres. And this has come from a model which is not data assimilating. It had been initialised 100 days earlier. So what we're seeing here is the evolution of the model state to be able to adequately predict a surface remote sensing reflectance. And we show this at 547 and also 667 nanometers. But like I said, we can do this at any wavelengths that we choose. The exciting then component is that we currently, this was a photo from yesterday of a research voyage that we've got underway in the GBR at the moment. The NASA Coral project is currently underway as well, and they have a multispectral sensor on the underside of the plane taking swaths up the reef. So they are able to produce in situ reflectances. So again, this is another data set that we can potentially exploit. And the other one is the fact that now that everyone has their mobile phones, you can get an app on your mobile phone which you can essentially get an in situ remote sensing reflectance from at the RGB wavelengths. And if we adequately couple that with a database, we suddenly have the opportunity for quite large scale citizen science activities. And not only can we collect the reflectances, but we can subsequently assimilate them back into the model using the assimilation methods. So assimilation has been proven to be particularly useful in NWP, and I suppose in many respects started off as a tracking problem during the space missions. And we've set it up so that we've got a 36-member dynamic ensemble. We know this is small and we're about to move to a 100-member. But we've undertaken a number of assimilation experiments where we look at assimilating the remote sensing reflectance or optically derived constituents. And we see that it's a bit busy, this plot, but just focus on the blue and magenta lines here. We can see that we substantially increase the skill of the model, especially with respect to TSS as compared to the control run. So this is the effect of assimilating reflectances as opposed to a chlorophyll product. And not only does it improve the estimates of chlorophyll and TSS, but we see that it also improves the estimates of the inorganic nutrients of nitrate, ammonia and DIP. This here is an animation of the 3-month assimilating run that we've got going. And we're about to set off a 6-year BGC reanalysis for the GBR in mid-November. The other thing being, now that we're predicting remote sensing reflectances, we can compare, so this is observed true colour with simulated true colour. So simulated true colour here has come from a control run. This is the simulated true colour from the assimilating run. And the way in which the assimilation has improved this solution is by adding sediments in this region here and reducing phytoplankton in the offshore regions. Okay, so this modelling wasn't all done for fun. It was largely done to try and assist with the management of the Great Barrier Reef. And one of the things that the model allows you to do is run a heap of what-if scenarios. So we looked at what would the improvement in water quality of the reef be if you took pre-industrial nutrient inputs and compared them to current day nutrient inputs. And for many of the inshore waters, it didn't actually make a huge amount of difference, which was surprising to some. But the reality is that for most of the inshore waters, the impact of catchment-related activities is constrained to a very narrow coastal strip. And what we expect is that the larger impacts of anthropogenic influences are going to be from the effects of ocean acidification and the increased storminess of the region. The other thing that we're hopefully contributing to in the near future is these marine condition reports. So these are traditionally generated using in situ observations and remote sensing, but now that we're about to commence a near real-time data assimilating model at high resolution, we would hope that we can use the modelling solution to contribute to these reports. So just finishing off on that, if we took the NOAA prediction of the coral bleaching event and then use the RECOM model to do that, and compare the severity of bleaching, so S being severe, M major, min being minor, we see that the RECOM model captured very nicely the observed bleaching event as observed by Ames. So in summary, the downscaling is useful for a number of reasons, primarily allowing us to increase the process complexity. We see that we can use this to quantify the ocean acidification impacts, and the fact that data assimilation, the way we've introduced the observations to the model, is able to reduce forecast errors across all the state variables that we've looked at so far, and that's a particularly encouraging sign. And the output of this model can be tailored to answer some very specific management questions. But sort of looking globally, what are the hot topics in the regional downscaling? Well, first of all, I would say that there's some very nice work being done here in France by, I think it's Florence Garnier, at stochastic processes. So it's a very nice way to include uncertainty in the models. I think we're going to see more operational high-resolution coupled hydrodynamic and BGC assimilation. And I think a good example of that is that work done by Chris Edwards' group. Improved DA algorithms. We know that ENKF, it's been around for 20 years now, but I think there's going to be some major steps forwards when we look at particle filtering approaches. And Peter Jan van Leeuwen has been championing that, at least in the hydrodynamic space. Model selection, there's various questions around complexity that would be nice to answer if we had better data sets. But I think bioargo and the inclusion of biooptics are particularly exciting advances in this space. So thank you very much, and happy to answer any questions.